Good afternoon. My name is Aaron Neal, and these are my group partners, Soyet Blue, Patrice King, and Raven Scales. And today we're going to be talking to you about neurotransmitters and amino acids. So I'm going to start by giving you a brief overview. We're going to start by talking about the overall nervous system, and then we're going to break it down to individual neuronal firing. Then we're going to go into neurotransmitters, their specific structures and functions, neurotransmitter receptor binding, the process of reuptake, as well as clinical implications and conclusions. So I'm going to start by talking to you about the central nervous system. The central nervous system is composed of two individual constructs, the brain and the spinal cord. As you can see pictured here, in the top right-hand corner is a brain, and then an even closer look is an individual neuron. And this can be seen from both the brain as well as the spinal cord have individual neurons. In the bottom right-hand corner is a picture of a neural network. So all of these neurons are going to communicate with one another through electrochemical potentials. And we're going to discuss that further in the next slide. This is a picture of an individual neuron. Now, the way that this electrochemical communication works is by each one of these individual neurons passing along an electrochemical message. So, say for example, this is the starting neuron. As you can see, this is just a regular cell. It has a cell body, a nucleus, and then it has a couple of other things that make it a little bit more individualized from other cells and make it a neuron. Now, the way that this electrochemical message would start is that sodium would rush in to the cell body as well as calcium, and it would start this electrochemical message. And then it continued this chemical message down the axon through these things called myelin sheaths. And these myelin sheaths are going to insulate the axon and allow this electrical message to pass all the way through until it gets to the axon terminals, where neurotransmitters are going to be released. So here is an up-close area of these axon terminals, and in between what's known as a presynaptic and postsynaptic neuron. A presynaptic neuron is the very first neuron, and the postsynaptic neuron is the neuron that's going to be receiving the next message. The presynaptic neuron, as you can see from this yellow lightning bolt, is going to release this electrical message all the way down to the bottom, where it's going to result in calcium exiting the presynaptic neuron. When it exits that presynaptic neuron, it's going to release these vesicles, as you can see here in these purple balls. And then inside of those purple balls are going to be these little red balls. Those red balls are going to be neurotransmitters, and those are the chemicals that we're going to be focusing on for today. This is an even more up-close version of it, and as you can see, these neurotransmitters are going to be in excess inside of these vesicles, and they're going to be released, and they're going to bind to receptors on the postsynaptic neuron, which is going to continue the electrical chemical message. Now, this is an example of a video that's going to show this entire process. So as you can see here, the sodium's going to rush in and the calcium's going to rush out, and that's going to continue this electrochemical message down the axon through those insulated axons through the myelin sheath, where it's going to release these neurotransmitters at the axon terminals to the postsynaptic neuron, where it's going to continue that electrochemical message all the way down to the very next axon. This is just on a smaller scale, but this happens on a neural network of billions and billions and billions of neurons. So next I'm going to give you a little more information on neurotransmitters. Neurotransmitters can be broadly classified into excitatory and inhibitory neurotransmitters. Excitatory neurotransmitters are going to continue the message down axons and down all the different neurons, and then inhibitory neurotransmitters are going to stop that message at the next axon. Neurotransmitters can further be broken down into three biochemical categories: biogenic amides, which include serotonin, histamine, and dopamine, as well as a number of other neurotransmitters; neuropeptides, which are amino acid linked neurotransmitters, and those include oxytocin, corticotropin releasing hormone, opioids, and about 100 other neurotransmitters. Just a fun fact, oxytocin is known as the love neurotransmitter. Whenever you're in love, there tends to be a high amount of oxytocin located throughout your brain, as well as your central nervous system. And this has been tested in mothers whenever they're bearing a child. There's a high amount of oxytocin because they already love their child. Amino acids are going to be the primary focus for our presentation.
Amino not associated with neurotransmitters, this being that they are not unique to neurons, and they are cellular constituents, and they are needed for protein synthesis. Antibodies that can be used to identify neurons, but there are no markers that use amino acids as transmitters. Amino acids produce compartmentalized substances used for making excitatory and inhibitory synaptic potentials. Amino acid neurotransmitters are all products of metabolism, except for gamma-aminobutyric acid, also known as GABA, which is glutamate's metabolic product and an inhibitory neurotransmitter. GABA is produced by an enzyme located in the neuron, and GABA is not used for protein synthesis. Glutamic acid, also called glutamate, an excitatory neurotransmitter. This is the structure of aspartate, which is another neurotransmitter. And this is glycine, the last neurotransmitter.
I will now talk about functions of neurotransmitter amino acids. I will begin talking about glutamate receptors. They are major excitatory neurotransmitters in the brain. Glutamic acid can regulate the activity of neurons once released from the synapse. Signals from neurons can also be altered by presynaptic glutamate receptors. Referring to this picture, we have a presynaptic membrane and we have our glutamic acid in these little vesicles. They move out into the synaptic region and bind to the receptor. And once it goes to the receptor, you will now be in the postsynaptic region. It'll interact more in the dendritic region. Moving on, glycine receptors are located in regions of the central nervous system, such as the spinal cord and brainstem. The function of glycine receptors is to mediate synaptic inhibition, regulate the excitability of motor and afferent sensory neuron, and processing visual and auditory signals. Now, looking back at the picture, we have a glycine neuron and glutamate neuron that act as co-transmitters. So here we have the glycine come out of the presynaptic membrane into an NMDA receptor. Lastly, we have GABA receptors. They are the main inhibitory neurotransmitters. These receptors can also be found in the central nervous system. The role of GABA receptors are to cause the membrane potential of neurons to deactivate the neuron. Referring to this picture, we have a presynaptic neuron and a postsynaptic neuron. Over here, we have a GABA A receptor and a GABA B receptor. So this is all that this picture shows. Next, I will talk about neurotransmitter receptor binding. Neurotransmitter receptor binding plays an important role in causing action potential. In other words, when a neurotransmitter binds to a receptor, more neurotransmitters are released. Action potential is what causes the neurotransmitters to be released. Referring to this picture, we have a molecule binding to a receptor protein. Later on in the presentation, we will learn more about what the significance of calcium and sodium has to this.
We will now go into further detail about the overstimulation of glutamate. So now I will show you a video of an actual neurotransmitter release at the synapse. This is a video that actually shows a neurotransmitter being released at the synapse. If you were to look at it, the yellow circle would represent the vesicle, as well as the top portion, the top dark blue portion represent the postsynaptic, as well as the bottom dark blue portion represent the presynaptic. And the inside light blue will represent our glutamate. As you can see, it is now being released into the synapse region, which will then connect to the postsynaptic region. So, an overstimulation of glutamate can cause or can lead to Alzheimer's disease as well as Huntington's disease. As stated before, glutamate is a major excitatory transmitter in the brain, as well as if there is an enormous release of glutamate, as shown in the video previously, it can cause an overstimulation, which can lead to such diseases. Next, we'll talk about glutamate receptors. So this picture right here is basically a simplified version of a glutamate receptor, as well as glutamate, the vesicles, the presynaptic, and the postsynaptic. So here you have the presynaptic region, which are vesicles that are filled with the glutamate, as well as down here, these U-shaped things, they are your receptors. And it actually shows some of them have binding regions, and they have the glutamate connected along with them.
Now we will go on extra emphasis on the reuptake pathway.
For this picture, it basically shows that an abundance of glutamate in the postsynaptic can lead to an influx of sodium along with water that can cause the postsynaptic to swell. Along with swelling comes a large sustained influx of calcium that may lead to hyperactivation of many calcium-dependent enzymes. Calcium influx through the NMDA receptor appears to be one of the keys to producing neural damage, specifically because the blocking of the activation of this receptor attenuates some of the neural injury following stroke. So we have portion A here. It actually shows a vesicle filled with glutamate, which are the little blue circles, as well as a vesicle being released into the synaptic region. Next we have portion B, which shows your 3 different types of receptors. And we have NMDAR, which is the one that's most commonly used with glutamate, as well as AMPAR, as well as NGLUR. For portion C, we have a glial cell here, and it actually shows where glutamate diffuses across the membrane as glutamate. So it then changes from glutamate into glutamine by the enzyme glutamine synthetase. As well as once glutamine is produced, it leaves the glial cell, and then it comes into the presynaptic terminal as glutamine. It turns back into glutamate by glutaminase, which is the enzyme that turns it back, and then it repeats the cycle over again. So for portion D, we actually have a close-up of the NMDAR receptor, and it actually shows the sodium and calcium channels that we stated beforehand. These sodium and calcium channels actually move the glutamate through. As you can see, there's a glutamate molecule here, and it shows that it will go through the channel. NMDA receptors, they fall in the class of ionotropic glutamate receptors, along with AMPA and kyanate. Also, NMDA receptors, they activate slower, and they have a higher affinity for glutamate. That is one of the reasons why we hear glutamate being acknowledged or used with the NMDA receptor. In conclusion, we have previously stated 4 different types of amino acids that serve as neurotransmitters: GABA, aspartate, glutamate, as well as glycine. So now we have, for the current work that is currently being done, we have analytical chemists who are trying to figure out if they can use different amino acids to serve as neurotransmitters. For example, we have D and L serine that can be produced. There's actually a study going on now. They found that the enantiomers of serine can actually be used as neurotransmitters, as well as the other 4 that were named. And this now concludes our presentation on neurotransmitters as amino acids.